Hi, welcome to this new video session. So here in today's session, we'll be discussing a non-compartmental analysis workflow for analyzing a drug-drug interaction study. So I've come up with an example of amlodipine. So over here, let me quickly show you the data set. So we have amlodipine um, along with an inhibitor and an inducer drug that was administered, and plasma concentrations were measured at steady state. And yeah, we'll be using this data to go forward. So let me click on done. And so again, before going, we just need to perform some basic transformations. So let's do that. So over here, I click on transform. So to show you the entirety of a data set, so let me just use a function in clean. So this is just to show you. You don't need to do this in your workflow. So I would just remove duplicates in subjects and show you the entirety of the data set. So we have amlodipine reference or amlodipine alone when it was administered, how plasma concentrations over time varied, and we also have amlodipine along with a CYP3A4 inhibitor, and along with that CYP3A4 inducer plus amlodipine. So three different groups, and how we have the plasma concentration data. So now what we can do is we can see the group N number, right? So amlodipine plus CYP3A4 inducer is group 3, CYP3A4 inhibitor plus amlodipine is group 2, and amlodipine alone is group 1. So in the first scenario, what we can do is just have two groups to go to start with. Let's make it simple. So let's filter, and we'll filter out group is not equal to 2. We'll just remove the inhibitor part. Let's work on amlodipine alone versus amlodipine plus inducer. And then so it is not group N, group, but it should be group N because that is what the numerical column is. Group has text. Okay. So now what I would do is add um, a derive. I'll add an interdose interval. So because I've measured at steady state, the application should understand till, or what is the frequency of administration until you reach the steady state. So basically I would mention it as 24 hours for this example. And yeah, now the data set is ready to go for analysis. Let's rename it and make it more simple. AMLO versus AMLO plus inducer. Yeah, so this is good. And we click on done. Let's quickly do the same for the inhibitor and keep the dataset ready, right? Click on transform. Let's do the same step that we did. Filter group N is not equal to, we did 2 previously. Let's do 3 now. Just sorry, I guess we removed. Um, let me check. Forgot. Let me. Yeah, we remove 2. Let's remove 3 now. Group N equal to is not equal to 3. Yeah, and next step is creating a derived column II equal to 24, and that's it. So now you have your amlodipine versus amlodipine plus an inhibitor, CYP3A4 inhibitor drug. Okay, so this is now ready. So we have now the inducer, wherein group 2 was not included, and an inhibitor where group 3 is not included. So now the dataset is ready for analysis. Let's proceed forward, and let's first analyze the effect of the inducer on amlodipine. Click on run a new task and select. Let's give a name DDI NCA inducer. Let's go to the next step. we'll select user dataset, click on next. So we have subject, time, concentration automatically mapped. We don't need the period column exactly here. So because there's a parallel group study, we have individual subjects for individual groups, separate groups, right? So I would just group them by uh, the group N, or which treatment did they receive. So basically 40 subjects received both amlodipine and a CYP3A4 inducer, and 40 subjects received only amlodipine. And next, let's look at some summary plots. So we have the plasma concentration profiles for both inducer and reference. Um, let's move forward, directly go to step 7 to look at some summary. So here, if we look at this at steady state, the Cmax and both when administered along with an inducer, so it does not make a huge change as such. But I won't start interpreting the data right now. But just wanted to show how you can arrive at the results. So, and also you can also generate um, a ratio table. So here you can click on add parameter table over here and choose table type as ratio. So we just saw a summary table, right? So we can just mention a table caption, effect of CYP um, inducer on amlodipine PK, and let's choose our uh, parameter. So I'll just choose now as Cmax, and I'll choose my grouping factors as group, and reference group is my just amlodipine, right? Amlodipine alone. So using this, I can easily get my geometric mean ratios and the confidence interval around it. So this way I can get my ratio tables also for each of the parameter. So yeah, so you see the ratio table is now added, and the analysis is um, complete for the inducer. The same steps you can um, redo to uh, analyze the inhibitor effect as well, CYP3A4 inhibitor effect as well. Let me choose the dataset, click on next, group them by the group variable, that is what treatment did they receive, and again 40 subjects in each of the group. Let's go to step 7, look at the table. I click on add parameter table, choose the parameter table type as ratio table over here. I'll say this as effect of inhibitor, choose the parameter as Cmax, group by group, reference group as amlodipine alone. So again, the same way how I created uh, ratio table for the inhibitor and the inducer, you can do the same thing to get to arrive at the output. So uh, click on next and click on complete to finish or arrive at the final result. So this way you can analyze the drug-drug interaction using a non-compartmental analysis workflow in PumasCP. This was a simple design, again parallel group design. You just need to uh, use a bioequivalence module if you have a complicated design. If you have a crossover design, let's say, bioequivalence module, like I showed in food effect videos, would help you to arrive at the results. So I hope this video helped you. If you have any questions, please reach out to support@pumas.ai. We'll help you resolve those. And yeah, thank you so much for watching.